Hello and welcome everyone. In this podcast, we're going to talk about eukaryotic chromosomes. We're going to talk about the structure of them. We're going to talk about the different parts of the eukaryotic chromosome. We're going to talk about the overall size of the eukaryotic genome, and then how this large genome is packaged into a cell. Remember, we said that E. coli, which we just talked about, they have a genome of about, so E. coli, a genome of about 4.6 million base pairs. Just as a comparison's sake, I can't imagine I'm going to ever ask you to memorize and recite these, but you should understand the general point I'm trying to make here. Fruit flies have about 170 million base pairs. So quite a bit larger. But a fruit fly is more complex. Humans, you would expect to have, well, we might expect humans because we think of ourselves as the most complex organism. Perhaps we think that. We have 3.2, roughly, billion base pairs. But we have to be careful. More doesn't always mean more complex or better or more adaptive. Sometimes these are just numbers. We know that some plants, which themselves are quite complex, and I might argue more complex than humans because of their various life cycles, but like a trumpet lily, for instance, they have 90 billion base pairs. And this last one I want to tell you about that I find really interesting are the amoebas. Amoebas are, in general, they offer a lot of interesting details that sometimes are hard to imagine or explain, but they have a whopping 670 billion base pairs. Yet, interestingly, the number of genes between a fruit fly and a human isn't that much different. Fruit flies have, roughly 13 to 15,000 genes, and humans, we have roughly 23,000 genes. It's hard to imagine that those extra genes can be made up for by this huge difference. It seems that we wouldn't need that much DNA. And the trumpet lily and the amoeba, their number of genes aren't all that much different than what we see in fruit flies or humans. And we'll come back to this point later on, but I think it's important to address now. And that is, why do we have, if we're just going to compare, say, humans with fruit flies, why do we have so much more DNA than a fruit fly? Yet the number of genes we have is not that much more than a fruit fly. Turns out that humans have more non-coding regions, which make up a lot of these repetitive sequences that we talked about with the bacterial chromosomes. We also know that in humans, and unfortunately we don't have much time to talk about this in this class, but they're really important, these transposable elements. These are pieces of DNA that have plopped into our genome throughout evolution. Most of them probably from a viral infection, but they have remained in our genomes, and not just human genomes, all eukaryotes. But in humans, we know that our genome is about 45% of these transposable elements. Oh, I didn't write elements. Depending on where these elements pop in, they can influence gene expression and the timing of expression, and this allows for various evolutionary events to have occurred. We're still learning a lot about these regions, but one of my pet peeves, and if you want to get on my bad side, you'll use this term, junk DNA. Do not use junk DNA. None of this DNA, this extra DNA that doesn't code for a protein, is junk. Not junk DNA. Growing up, my mom had a picture on the refrigerator that said, "God don't make no junk." So that's what I think about here. There's a reason we have this DNA, and what we're discovering is that this DNA that many decades ago we called junk DNA, we are now realizing that it has a really important function, and by and large is what makes humans different from fruit flies, and fruit flies different from E. coli. These non-coding regions are really, really important. Okay, I'll get off my soapbox now, and let's start talking about eukaryotic chromosomes. Now, on the previous podcast, I drew a picture of a bacterial chromosome to illustrate some of its features. And I want to do the same thing here now with a eukaryotic chromosome. And we'll see some of the differences in eukaryotic chromosomes compared to prokaryotic chromosomes. The first thing that'll pop out to you is that eukaryotic chromosomes are typically linear. You will find some exceptions to this rule, but in general they are linear. In a typical eukaryotic cell, you will have multiple chromosomes that have different genes on them, and so we'll have multiple as an example, humans, we have 46 chromosomes, or we might say we have 23 pairs of chromosomes. Because that brings us to our next point about the multiple chromosomes. Remember, we said with bacterial chromosomes, you only have one chromosome and it may be present in one or multiple copies. In most eukaryotic cells, we either have one copy, which would be a haploid, as we've talked about before, or in a diploid state, where you have two copies of each chromosomes. Humans, our cells, most of our cells except the sex cells, are diploid, and so we have 23 pairs of chromosomes for a total of 46 chromosomes. Different organisms will have a different number of chromosomes and different number of pairs of chromosomes. Another point to make out while we're talking about this is that the number of chromosomes, just like the number of base pairs, didn't signify anything about the complexity of the organism. The number of chromosomes also don't say anything about the complexity of the organism. In eukaryotic cells, we have multiple origins of replication. And again, my abbreviation for replication. Roughly speaking, these origins of replication occur about 1 in every 100,000 base pairs. And so let's go ahead and draw them on here. These are just approximates. It's important that we have multiple origins of replication in eukaryotic chromosomes because a eukaryotic chromosome will have anywhere from 10 million to hundreds of million base pairs. It is necessary to have multiple origins of replication to copy this efficiently so that we can accomplish this within a matter of a day or so, as opposed to it taking months or years to copy the eukaryotic chromosomes, because they are so much more massive than a bacterial chromosome. The next feature I want to talk about is something called the telomere. Eukaryotic chromosomes have these. They are repetitive sequences, but bacteria don't have them. We only see them on linear chromosomes. And they're on the ends here, and I'm going to color them in purple. On the very ends. Okay, as I said just a moment ago, these telomeres are only found on linear chromosomes in eukaryotic cells. And I want to tell you a little bit about their functions. They're very important in stability. In most of our cells, every time the cell divides, the chromosomes get shorter and shorter at the ends, at these telomeres. At some point, they reach a critical size at which the cell is signaled to die through a process called apoptosis. So these telomeres almost act as fuses for the life of a cell. As they get shorter, the cell gets closer and closer to the point where it will undergo programmed cell death. So they're important in maintaining the stability of these chromosomes and the life of the cell. We'll talk a little bit more about these telomeres when we talk about DNA replication. Next, we should talk about a structure we've already talked about, but the centromere. Remember, we talked about that as a way that we classify chromosomes. In this case here, the centromere is right here. It's right in the middle, so we would call this a metacentric centromere. And the centromere is important in chromosome segregation. They're important in chromosome segregation because at the centromere we have a group of proteins called kinetochore. Kinetochore proteins. And I'm going to draw them in orange here, just give them a different color. But what these kinetochore proteins do is that they will bind to the spindle fibers during mitosis or meiosis. That's important in pulling the chromosome towards one side of the cell or the other. So these kinetochore proteins bind at the centromere to help in chromosome segregation. Now there's a few more things I want to talk about with this eukaryotic chromosome, so I'm going to erase what we have here and start with number 8. I'm going to draw these little dashes here to signify genes on this chromosome. And what you'll notice is that there are more regions in between genes, or I should say more sequences in between genes, and more repetitive DNA, which I'll draw here as pink. In fact, so this is going to be our repetitive DNA. And this telomere is also repetitive DNA. So I'm going to extend that through our purple dot there. We always see a lot of repetitive DNA at the ends and near the centromere, but we also see it throughout the chromosome as well. As I mentioned earlier, these repetitive pieces of DNA have an important function in regulating gene expression, folding the DNA, maintaining the stability of the DNA. Most of this repetitive DNA is likely inserted by viruses throughout evolution. Typically, organisms that are more derived, perhaps more complex, we could argue the complexity of an organism, but certainly those that are more derived, with more derived features, will have more of this repetitive DNA. Let's go ahead and erase this. Now, when we talked about bacterial chromosomes and how it was packaged, we talked about forming these loop domains with these DNA binding proteins and then how it's supercoiled. Eukaryotic chromosomes have a similar task at hand, but eukaryotic chromosomes have a different problem to face, and that is that they have multiple chromosomes that need to be packaged and compressed into a tight spot. Now I'm going to talk about humans now, but similar problems exist for other eukaryotic cells, and all eukaryotes use a similar solution. But I'm going to talk about humans just because I find it remarkable that within each of our cells, so a human cell, I'm just going to draw it like so, will have a nucleus, and that's another problem that the eukaryotes have, is that they have to do all this within the confines of a nucleus, so a smaller part of the cell, and they fit into this cell here, this very small nucleus, about 6 feet or about 2 meters or 2 yards of DNA. So if you can think of a person you know that's 6 feet tall, clearly not me, but other people who are 6 feet tall, and think if you put a piece of DNA at their toe and stretch it all the way to their head, that's how much DNA has to fit inside of this tiny nucleus. If you take this, another example that kind of shows the enormity of this process, is that the smallest human chromosome stretches out to about, if you stretched it out from end to end, about 14,000 times the length of a nucleus. So a nucleus is about, it's about 5 to 10 micrometers. The smallest chromosome has to fit into this 5 to 10 micrometers space, and it's 14,000 times the size of it. And that's just the smallest chromosome. In humans, we have 45 other chromosomes to contend with. And all of that has to be shoved in here. Similar to the bacterial problem, it has to be placed into this nucleus in a manner where it can be used, where it can be replicated, where it can turn on or off genes. So it's just not shoving it in there but it's placing it in there in a very controlled and organized manner. The way that humans first do this, and we're just going to look at one chromosome or one piece of that chromosome, is in multiple layers, similar to what we would see in the bacterial chromosome packing. This first layer of packing involves these proteins called histone proteins. And there are typically 9 histone proteins involved in this. We have 2 H2A, which stands for histone 2A proteins, plus we have 2 H2Bs, plus 2 H3, histone 3 proteins, plus 2 H4 proteins. Then we also have 1 H1 protein. This first group, these 8 proteins up here, they make up what we call the histone octamer. And I'm not going to be able to draw this three-dimensionally, at least not very well, so I'm only going to draw 4 of them. But there are 4 on the other side as well. And what this will do, we'll take the DNA and wrap it around these histone proteins. So I want to make sure I label this correctly. These 8 proteins, these 8 histone proteins, make up the histone core, or sometimes books will call it the histone octamer. The DNA will wrap around this histone octamer, and at this point, histone proteins with the DNA here, we call this the nucleosome. This nucleosome is very important in packing the chromosomes. It's also, as when we talk later on in the semester about gene regulation, we'll see that these histone proteins are incredibly important in regulating gene expression. But you'll have to come back later in the semester to figure that out. We're talking about packing now. And so let's go ahead and continue this out. We'll have another histone octamer here that the DNA will wrap around. We'll have another one here, and so you get the idea here that it will keep doing this for the length of the chromosome. The histone 1 protein I haven't drawn yet. It's not part of the octamer, and some species, like yeast, don't have the H1 protein. But it is presumed to have a role outside of the octamer, outside of the nucleosome, and it functions in stabilizing the overall nucleosome. In between these nucleosomes, we have proteins that will bind to this area here, this space in between nucleosomes. And these have an incredibly clever name called non-histone proteins. Now, before we talk more about these non-histone proteins, let's talk about the sensitivity of the DNA. So as I said, the DNA that is wrapped around here helps in folding the DNA, but it also prevents enzymes from coming into the DNA. And that gives you a little idea of how they're going to help regulate gene expression later on in the semester. But enzymes, if they were present, so if enzymes are present, and let's just draw an enzyme here, it could not get access to this DNA. However, enzymes have access to these regions in between the histones. As a result, enzymes like DNase, those enzymes that degrade DNA, would be able to degrade the DNA in between these histones. And experimentally, if you isolate chromosomes that are in this nucleosome structure here, the DNase will degrade these, and if you throw this piece of chromatin into a tube with DNase, what you end up with are essentially these linker pieces of DNA gone. So the linker here is gone, and you just have these beads, essentially, is what they would look like. The beads of the histone octamers present with the DNA associated with it. But all the linker DNA is gone, which tells us that enzymes have access to this area of the chromosome. Now you may ask, why do the histone proteins bind to the DNA? And it's important to remember, when we talked about the structure of DNA, we talked about those phosphate groups at the 5' ends. DNA, in general, has a negative charge because of the phosphate groups. Because of these negatively charged phosphate groups, these histones have an overall positive charge. So the histones, I'll put a plus sign here in green, and then the DNA is negative. And so the two will form this non-covalent interaction. So that concludes the first level of eukaryotic chromosome packaging. This is the most important part. This is what I'm going to expect you to know fairly well. But let's briefly talk about what happens beyond this nucleosome structure. Because remember, we still have to put 6 feet of DNA inside of a cell, so this only accomplishes a small part of that. And this is what I'll expect you to know. That we have kind of our naked DNA here. The first level of packing will be, I'm going to draw this a little shorter, will be to compress it some using these histone proteins forming the nucleosomes. So the first level, as we said on the last picture here, is nucleosome. After this, the nucleosome sort of brings the nucleosomes sort of begin to fold upon themselves. And I'm not going to be able to draw this very well, but you can look at the image in your book, because this will form what is known as the 30 nanometer fiber. And it's not going to look like this over here, but it's going to appear in such a way that the nucleosomes are almost like right on top of each other, because they're folding upon each other. And much like the loop domains in bacterial cells, eukaryotic cells will next form what's known as radial loop domains. And now I'm going to draw a much larger piece of DNA beyond what we see here. And essentially what we're going to have here is proteins that we call scaffolding proteins here. And these loops of DNA that are in the 30 nanometer fiber form will now begin to fold up on it, attaching to these scaffolding proteins. This is also called the 300 nanometer form. Then what happens is that these loop domains that are attached to these scaffolding proteins will further compress. And it mainly does that by the scaffolding protein kind of folding upon itself. And as it folds upon itself, it's also further compressing the DNA that is attached to it. And we would call this the 700 nanometer form. The last form that we have is as this further compresses, then we eventually will see what we would call that chromosome structure that we see during mitosis or meiosis. And it's called the metaphase chromosome, where it's perfectly compacted to the point where what you really see at this point is just, and you can see this under a microscope pretty easily. I'm going to have to draw it up here just for space reasons. But it's when that chromosome is now perfectly compacted and ready to be segregated during mitosis or meiosis. So we're going to call this the metaphase form. So this, I think, is rather complicated. And as I said before, this is where I'm going to want you to put most of your attention. Understanding how these nucleosomes form and why they're important. Beyond that, I think it's important to know the order of these structures. That you go from naked DNA to nucleosome structures, to the 30 nanometer fiber, to the 300 nanometer fiber, to the 700 nanometer fiber, and then ultimately to the metaphase chromosome. It's useful to draw these to give you an idea of what they go through, but I can't imagine asking you to draw anything to this side of my pen on an exam. This side of the pen, I could see asking you to do that. But you should know the order here. And you should know that this is how eukaryotes package 6 feet of DNA into a nucleus that is only 5 to 10 micrometers. Okay, there's two more terms that we need to talk about and compare, and that is the term Euchromatin versus Heterochromatin. We think of Euchromatin as being dynamic. Most of a eukaryotic chromosome is going to be euchromatin for most of the time. Euchromatin is less compacted. Heterochromatin, we would say, is very stable and highly compacted. Euchromatin is transcriptionally active. And just to remind you, transcription is the process of making RNA, which then can go on to make protein. So this means that the genes in this area would be euchromatin, where genes are being turned on. Whereas heterochromatin is transcriptionally inactive. You would find euchromatin in areas where there was active gene expression. You would find heterochromatin in regions of the chromosome that are not being actively transcribed, that are kind of, for lack of a better word, kind of inert. These would include the regions of the centromere and around the centromere. These are very gene-poor regions. The telomere is always heterochromatin. The Y chromosome is almost all heterochromatin because there's very few genes on the Y chromosome. And something we haven't talked about yet, and we won't until near the end of the semester, but one in females where you have two X chromosomes, one of those X chromosomes is inactive. So the inactive X chromosome would be heterochromatin. It's also important to point out that you could have a chromosome, and I'll just draw it like so, where parts of it, and I'm going to just draw it as a kind of a squiggly green line here, parts of it are heterochromatin around the centromere on the ends, but the rest of it is euchromatin, where we have active genes. So a chromosome isn't always either all euchromatin or all heterochromatin. Usually, particularly as the cells are living, it will be a mix of both euchromatin and heterochromatin. The only time you would find a chromosome that is all heterochromatin, so it might look something like this, is during mitosis or meiosis, when that chromosome is completely packaged up into that metaphase type chromosome. Now that ends this podcast on eukaryotic chromosome structure and packaging. Let's do a quick summary here. What you should have got out of this podcast is the structure of eukaryotic chromosomes. So remember, we talked about the different parts of it, including the centromere, the telomere, the areas of repetitive sequences, the kinetochore proteins that bind to the centromere, the multiple origins of replication, and that the overall size of the eukaryotic chromosome is much larger than that of the bacterial chromosome, in a large part due to the massive amounts of non-coding DNA, such as the repetitive sequences. We then talked about how we had to package 6 feet of DNA into a tiny nucleus. And we talked about how it did this by going into great detail about the structure of the nucleosome. Remember, that was DNA plus histones. And then how this nucleosome was further packaged through multiple steps into the 30 nanometer form, the 300 nanometer form, the 700 nanometer form, and then ultimately into the 1400 nanometer form, which was the metaphase chromosome. So be familiar with this process and very familiar with the nucleosome. And then lastly, we talked about the differences between euchromatin and heterochromatin. Well, that's all I have for this podcast. If you have any questions, please make sure you let me know, and I'll be happy to address any questions you might have. I'll see you in class. Bye.